so very much. It's always a pleasure to be here. These events are really fantastic to connect the patients and the recent knowledge. So please, as Dalton said, get any questions in there. We will be having some time to discuss them. So let me start, go off what happened in MPNs in 2025, what will hopefully happen and will continue to happen 2026 and beyond. So if we can go to the next slide. I would like to take you to the journey of MPNs of 2026 and next, which hopefully will be a new targeted era of myeloproliferative neoplasms. So what's worth watching, what's coming on, what's happening in there? It's quite fast-moving field, and I will spend mostly time on the novel therapies that were presented last year at the major conferences and all of those that we have questions about and can enroll patients on if needed. So one is the CALR-targeted development, which are the antibody that will probably be the growth of the topic, but then also expanding indication of RAPEG interferon from polycythemia vera to ET. And then in myelofibrosis, I'd like to mostly focus on the novel approaches or the longest data on combinations. We all are aware we have 4 JAK inhibitors approved for myelofibrosis in this year, but they all approved in a single agent in line agnostic fashion. We have not had any combination that made it to the approval yet. So I will give you some data on that, and then we'll be discussing all of the new updates. So let's start on the next slide with the CALR-targeted. So as you all probably aware or heard of, what is CALR-targeted? It is a mutation that happens in up to 30% in patients with essential thrombocythemia or myelofibrosis. And then what it does, it basically creates a new, what we call C-terminus. You can see a little complicated slides from scientific endpoint here, but bottom line, CALR-targeteds are the drivers that would induce the independent signaling and cause myeloproliferation. We have discovered CALR-targeted as the last driver in 2013, and only in the very recent years, we have found out how does it work and whether we could target it. And here we go. This is the first data that were presented for humans that I will focus on something called INCA 909. I will make it a little simple so you can follow me, but bottom line, it's a monoclonal antibody. So it's a molecule created to selectively bind when it's given intravenously to patients onto that mutant CALR-targeted molecule that is at that time already bound into the receptor that is responsible for the molecule to cause the independent signaling and growth of the ET or MF cancer cells. On the right box, there is a little bit cartoon showing the development of the INCA antibody. And then how does this work? Again, a little bit scientifically profound, but bottom line, we were able to discover that when this mutation happens, that drives ET and MF in 30% of patients, it travels from inside of the cell on the surface. There we could basically target it, develop an antibody, which is this one, bind it to it when it's given to patient, and it'll on its own kill the cancer. That is actually an approach that's been used across different diseases over the past couple of decades. We have never been able to do similar things here until we understood that we have one immunogenic target that's mutant, CALR-targeted. So let's dig into the data. If you, Dalton, can show me the next slide. These are the first data of the INCA 909, again, presented from humans. So 2 clinical trials that were run across the United States or outside, mostly in Australia, included 2 patients group. 1, ET patients refractory to frontline therapy and high risk. Other myelofibrosis patients, either monotherapy, so after failure of JAK inhibitor, which are current standard, or add on to only one JAK inhibitor, the ruxolitinib, the first, that was on the far right, only study run in Australia, not in the United States. And it added the molecule to stable dose of ruxolitinib for patients that would not have ideal response. So here you can see summary of patients enrolled. Again, first data, but already pretty good cohort. ET patients 55, myelofibrosis 52 for monotherapy and 20 for combination. And then I'm not going to go and show you all of the details. You can see them here. But bottom line to highlight, ET patient needed to be high-risk refractory. So median platelets when they started were 1 million. So pretty significant patient population. In the myelofibrosis, the things to highlight, significantly symptomatic and large spleens. The spleen volume, which is how we measure the size, that should be less than 300 cubic square. You can see on the far right is over 2 liters. Huge spleens. So now let's see what we had next. So it's a phase 1 study. So we are just trying to find out whether that works and whether it's safe. So the primary objective of early studies are always safety. We have to prove it doesn't cause any harm. And that has been met. It's highlighted on the left upper part, checkbox. We've shown it's incredibly safe molecule. It is given currently on the study that I am presenting intravenously every 2 weeks. That has been changing, but it's not been implemented yet. But this is the data I will share with you. So far right on the box, you can see the adverse events that were reported on the study specifically related to the treatment. The T is treatment-associated adverse events that would be deemed to be therapy-related. Serious adverse events are any very significant ones we have to be concerned about, and grade 3 would count into the same. What's important is the discontinuation or dose reductions that's highlighted in the middle of that table with the red box. So adverse events that were attributed to therapy that would lead to treatment discontinuation reduction will happen only in 1 or 2 patients respectively on each of this cohort. That's actually a remarkable finding for phase 1 clinical trial that never had any data in humans. On the left, basically speaking towards how safe this molecule was, is patients that remained on the study as of the last follow-up, and you can see it was 85% with combination to ruxolitinib with a very advanced patient to 93% of patients on ET only. So almost everybody stayed on the study, and there was no toxicity dose that would be concerning or limiting. That's highlighted at the bottom on the left. No dose limiting toxicity. That means we met the endpoint we wanted to show. On the next slide, I will share the first preliminary data, why we are getting excited about this. On the left is ET, on the right are myelofibrosis. So because it's a phase 1, it has a dose escalation. We are trying to find the dose that's going to work for patients, and it has a very broad starting point, only 24 milligrams, but the range is 2,500, so quite broadly. And then on the left, you can see the hematologic response, simply meaning control of platelets or other signs of disease in patients with ET. And again, over 80% of patients going to as high as 90% of ET would have control of the platelets and other hematologic parameters. If you can see the platelet count graph, I know a little complicated, scientifically proven, but it has platelets number. And as I told you, every 2 weeks intravenous, the second bar is platelets almost going to 500, starting for over 1 million on the left. The 1,000 equals 1 million in our measures. The second dose would be already less than 400. So patients' platelets could drop after just 1 dose to almost normal levels, then drop normally and remain low as patient continue. That's actually absolutely fantastic. On the far right are standard criteria for myelofibrosis as are currently expressed, and I will talk about more throughout this webinar. SVR 35 or TSS 50. So those are achievements of reduction of spleen volume by 35% by imaging or symptoms by 50% on the scale that's measurable. You can see across different endpoints, either 35% reduction, 25% reduction, or best reduction were all at least at observed levels across the studies currently, so 35% or more. What's interesting is also improvements of anemia. That was evaluated in monotherapy or combination with myelofibrosis. And the most exciting was 40% of patients, monotherapy, refractory, failing JAK inhibitors with myelofibrosis would achieve transfusion independence. That's actually very exciting and was not even initially thought this molecule could do it. But here we go. We were very happy to see this. Next slide will show you a little bit more details about why is this important. So here we go into not primary but secondary endpoints. So they are just telling us how deeper we could go. And this is reduction of calreticulin. You notice I'm talking about anti-calreticulin monoclonal antibody that's being given to patients by infusions every 2 weeks to go in, lock into calreticulin, get rid of it. The bars dropping below the 0 on left and right in the figures, you can see left is ET, right is myelofibrosis, are decreased percentage of the calreticulin from starting point. So they start at certain number, which is expressed as 0, but they could be anywhere from 5% to 30% or 50%. And then what is the percentage of decline as of last measurement? And you can actually measure to our patient that the carleticulin would decrease more than 90%. I have to first remind you here, this is very short follow-up. The study started to enroll only a year prior, and median time, which is the average people were on, is not even a year. So this is actually fairly remarkable. We have never seen a molecule that would decrease carleticulin burden this quickly and to this level. And the right upper part in the table are summary of any reduction. VAF means variable allele frequency, which means the carleticulin percentage that we could measure in the cell. How it dropped, 96% in ET patient would have some drop, 89% in myelofibrosis. That's wonderful. And then measurements that would be more significant drops, but with the short time, this is actually very, very positive. Next slide is even speaking and talking more about this. This is a very deeper looking at the bone marrow. On the left part, it's bone marrow biopsy under the microscope that is being colored or stained for the mutant cells, particularly platelets precursor, because platelets go with ET or myelofibrosis, and their abnormality is mandatory for diagnosis. So we know with the carleticulin mutation, the platelets express that marker on the surface. That's shown on the left upper corner with the yellow dots. The yellow circled cells are mutant carleticulin carrying megakaryocytes that will rise into mutant carleticulin disease targeting cells in the patient's blood. On the bottom of the same picture, the CD71, which is a marker for megakaryocytes, is showing more of the blue and less of the yellow, which is then also translated in the middle of this slide, showing left ET, right myelofibrosis, patients that would have repeated biopsies and measurements of how do these megakaryocytes or platelet precursor color before start, on therapies, and at the last follow-up. You can see the middle bar, the yellow highlighted one, is decrease of the mutant or sick carleticulin mutated cells down. So almost 80-90% decline in seeing them under the microscope, as well as with special staining. And what's actually more exciting to me and all of us watching this is on the right part of that. You can see that there's a little blue bar, but that blue bar is actually formation of new healthy carleticulin negative megakaryocytes. So not only are we already destroying the bad one, the yellow ones, we are allowing opening space of the bone marrow and the rising of healthy cells that would give us happy platelets. That means that's why we haven't seen significant toxicity in people having too low platelets on the antibody, because as we are destroying the disease-driven cells, we are already seeing growth of new ones that don't carry the marker, which is absolutely fantastic. More of this is also highlighted on the far right in the red box. It's red because it's actually all the excitement that I can see here. And it's telling you not only we have seen this in platelets, we have seen this in the red cells as well. We have seen this in something we call HSPCs. You can see the abbreviation on the right, hematopoietic stem cell progenitors. So ET and MF and all MPNs are hematopoietic stem cell diseases. They start within the bone marrow. It's a disease that's embarked within the cell, and then the initial sick cell that carries carleticulin then gives rise nonstop constantly to these abnormal sick cells. So we have seen actually decline of that initiating cell, which means we might have an ability to completely eradicate this and cure it. I think that's the red box of this whole Inca carleticulin. Very exciting, finally seeing behind just controlling of the symptoms and spleen, we see actually changes on the molecular level, declining carleticulin burden. That is a consequence of mutant cells, and we are seeing the cells to go away. I think this is going to be something to watch. There's definitely going to be more people, more studies to evaluate this, and then we'll see where it can take us, hopefully to completely change the whole landscape of what we call now is still ETMF with carleticulin mutation. I'm hoping it will be called only carleticulin defining myeloproliferative neoplasm that will be exactly targeted to be cured. What about we talk about the next topic? That's interferons. So raw peg interferon, and I've seen some questions coming along. I'll be happy to discuss them. I am not ignoring you, but let's just go real quick. Keep them in your mind. We will address them. RAW-PEG interferon, it's interferon, the only molecule approved in PV, so polycythemia vera. That was approved in the United States in November of 2021. On the left side, you can see the latest results of the study at 7 years from this initiation already had approval that show that interferon is another drug that could not only control the blood counts, but could decrease the allele burden, at that time only measured by JAK2. So now we switch in the gears, coming from calreticulin drivers to JAK2 drivers. They are noticeable in about 60% ET myelofibrosis and about almost everybody in polycythemia vera. And at the bottom of that slide, you can see actually the decrease of the JAK2 burden, something similar you've seen before with calreticulin, but now it's comparing the not treated patients. So the level of achievement of reduction of the burden of JAK2 was significant. Actually, up to 22% of patients in this study after 7 years achieve undetectable JAK2. So we were not able to detect the disease-initiating clone in the blood. But I'm going to focus on the right and the following slides for ET. ET doesn't have RAW-PEG interferon approved. We had interferon that's called pegylated interferon, which was one next generation that was a little bit less clean. The RAW-PEG interferon is monopegylated, very clean, one typical substrate molecule that is given subcutaneously every 2 weeks in a maintenance setting every month, less frequently, and the side effects profile is much favorable, or at least seem favorable to all the older interferons. Why interferons? They also have ability to target the disease clone that I just elaborated so profoundly on calreticulin. So what data we have this year? If we can show the next slide, Dalton. We have data in second-line setting of ET patients. So again, approval is in PV. Now we're focusing on ET and broadening the indication. Relapsed refractory patients with ET that failed one therapy before, it didn't have a name, any therapy, and then had to be intolerant hydria in the first setting. So at least one had to be hydria, and then having high-risk disease. Randomized 1-to-1, you can see on the right, the green RAW-PEG interferon compared to anagrelide. Anagrelide as a standard second-line setting in refractory ET patients. And then at what we call primary endpoint, 52 weeks, we were looking what happened to these patients. So next slide gives you the endpoint, which was control of hematologic parameters, no progression of spleen and symptoms or the improvements. Then it's quite clearly significantly different. With the green bar, RAW-PEG interferon significantly improved control of this disease versus anagrelide shown on the right, almost not even visible. And then more importantly to me, on the far right, if you look at the little bit more reddish box, there are individual parameters improved by interferon versus the anagrelide. And you can see that all of them would be at least twice as controlled, if not 3 times, with RAW-PEG interferon versus anagrelide as the current standard of care. So this study met its endpoint and showed that in the second-line setting, RAW-PEG interferon should be used ahead of anagrelide, which has been established care for the past decades. Next slide is showing you safety. Interferon's always been safety concern for why we don't use it that often, but here you can actually see the safety on the study at that endpoint. The whole side effects profile shown on the left. On the right, the more significant concerning, again, named very similarly, treatment emerging adverse events, TEAI, either very severe, leading to discontinuation or drug changes. You can see again, anagrelide is actually bold on the right because it was twice as side effects profile that RAW-PEG interferon. So the interferon was twice as good. So here again, we confirmed that the fear not using this agent in a second-line setting shouldn't be there. It is safer, more tolerated, and better effective molecule for refractory ET patients. The next slide will close the gap on ET and PV and move into the myelofibrosis space, where I would focus on the combination. This one is a study that has been around for some time, Manifest-2, that was basically the largest study in myelofibrosis so far reported with this extension. This is longer-term follow-up. So JAK2 naive patients randomized to pelabresib, ruxolitinib, or ruxolitinib placebo. So ruxolitinib, very well-known, approved, FDA-given JAK inhibitor, was either doing with placebo or people received something called pelabresib, which is a bromodomain inhibitor. Novel molecule that should go after the disease initiation and give us better results. In the first read of this study, we have seen significantly better control of spleen control, twice as better, as well as dual, but we haven't seen significantly better symptoms. That's why this study didn't meet the approval. And now what I'm presenting to you is update at 96 weeks. So if we show the next slide, you will see the responses achieved over time. On the left is week 96 of spleen volume control. Again, very similar bars before. The drop from 0 by certain percentage is improvements of the spleen volume measured by repeated imaging at the week 96, so almost going to the 2 years mark. On the right is the same at symptoms. So the orange color would be patients that were only taking single agent ruxolitinib plus placebo, and the blue overlapping and going down would be taking the combination. So here you could see that it showed sustained, ongoing, and unreached. So we don't know what the median time of the combination will be for both of these controls. On the next slide, we can see that there was also improvements in anemia that's shown on the left. The bars are showing baseline or repeated hemoglobin levels over time going until the week 96. The blue population are patients that were taking combination. So better control and less impact of the anemia than on the ruxolitinib only that was expected. So here we could extrapolate that the combination does improve hematopoiesis and doesn't cause the anemia that often. On the right has been incidence of leukemias that has been concerning up front, seen in the first readout, has not shown to be much of a significance because the curves at that time look the same. That is actually a backbone for Manifest-3, which is a study that's launching. We have a little bit different eligibility, but kind of similar design to prove a concept whether the combination of pelabresib ruxolitinib is beneficial to patients or should be abandoned. Next slide. We'll continue with the space of combination in myelofibrosis, but introduce new molecule concept called elritercept. Elritercept, you might have seen or met something called luspatercept that has been used in myelofibrosis or disease called myelodysplastic syndrome that improves anemia. Elritercept is a novel molecule that works through cytokine, the little bit complicated mechanism, but bottom line, it does decreases the anemia-driven signaling and improves when patients being transfusion dependent. So the study had a little bit a complicated design. Part 1, part 2, dose finding, as again, every early phase needs to first define the dose. And then I'm presenting combination with ruxolitinib, where the elritercept has been given on top of stable dose of ruxolitinib. So it's a concept that showed could additional medication in patient that would be anemic on ruxolitinib stable dose improve the outcome, primarily looking, could that be less transfusion dependent? On the left, you can see the safety. Again, early phase, safety has to be shown, quite toxicity acceptable, not many discontinuation because of severe adverse events. On the right, you can see that most of these adverse events were hematology, so decreased blood counts, which the patient started with. This is not what happened on a study. This is any observed. Because patients entered the study with low counts, they are definitely reported. With the next slide, you can see the efficacy in controlling the anemia. That's the bars on the top with the gray colors. And on the far right is a summary of transfusion. So RBCs are red blood cells. TI is transfusion independence. So more than 40% of patients that were dependent on transfusion, that means at least 6 transfusions a month, achieved complete independence when they were added to ruxolitinib. Moreover, patients that would have improved at least 50% decline. So let's say you needed 6, now you need only 3, had almost 65% of them. So significant improvements in transfusion requirements, dependency, and at the front of the slide or the top shows a different time point. 12 weeks, 16 weeks, 24 weeks, how it lasted so long, or overall including improved hemoglobin. That was actually very exciting. I think what was more exciting was the bottom part. We have not only seen improvements in anemia, as the drug is designed to do, we have seen significant more improvements in size of the spleen and symptoms. So that was not what the drug was designed to do, and that was positive. If we improve the transfusions and the red cell status, we do see patients feeling better and having even smaller spleen, maybe because we calm down a little bit of the requirements of the anemias. So that was positive. Watch out for the readout of this study because it will be continuing maturing. The next one is a novel concept of something called momelotinib, which is the latest approved inhibitor in the United States, just 2 or 3 years ago, with nuvisertib. Nuvisertib is something called PIM kinase inhibitor. So a lot of wording, but bottom line, they work at the inflammatory background, and they are supposed to synergize and deeper control not only the inflammatory part of the disease, but also the disease-initiating clones. Again, complicated study, having dose-finding portion on the right are different doses that were used of the nuvisertib, that's our all bulk pills, added to momelotinib. There were no dose-finding toxicity, so the highest dose proposed actually became the evaluated dose, which means the drug was very safe. The criteria are shown there. It's refractory because it's a next combo. And then the box in the bottom shown safety. We have to show it again. I'm repeating myself because I'm bringing phase 1 studies, but quite safe. The most concerning were gastrointestinal events, but they were fairly manageable. They were mostly grade 1, grade 2, and needed to be supported, but usually lasted 1-2 months and then disappeared. And then importantly, there were good responses that I've seen, and I was excited about the bottom, adiponectin or the inflammatory markers, significant correlation between seeing some improvements as well as declining an inflammatory marker and improving an anti-inflammatory marker. So the nuvisertib add-on basically work for the purpose of changing or deregulating the inflammation in these patients in terms of giving us better responses. And you can see the next slide, which will probably about to finish, would be the standard spleen and symptom responses on this combination that tells us, yes, we see reduction in the spleen as well as the symptoms. Symptoms are on the left, the TSS total symptom score, and on the right, spleen volume reduction, where you can see the combination in refractory setting when added to momelotinib brings us with better disease control. So, and I think we are about to finish here with our talk. If Dalton can move it to the next step, I will just give you a brief excitement about what to watch for 2026, 2027, and now on. I haven't had time to talk about hepcidin mimetics or other agents regulating iron metabolism. That's another big area of evaluation that will come into polycythemia vera, patients that would be dependent on phlebotomies. The first 2 bullets include one drug called rusfertide. You might have heard about it. It's the most developed, has mature data of phase 2, phase 3, and should hear about FDA status this year. So that's the last one that showed significant improvement in phlebotomies, and the latest in the development that may get approval. The next is sapablursen, which is a very similar molecule, but doesn't work that way, works on negative regulation of the hepcidin. So very similar concept, but just a different level. That has a phase 1 and met criteria. The next 3 molecules are the same level of doing something with the inflammation or signaling pathways in MPNs, bomedemstat or LSD1 inhibitor that has data now in ETPV, navtemadlin, which is the MDM2 inhibitor for patients with myelofibrosis, a different data at phase 2, phase 3 studies, currently a large phase 3 study that is still ongoing and enrolling. And then selinexor or export inhibitor, which works with the signal molecule, how different inflammatory markers and all kind of other enzymes are being processed in humans' bodies, added to ruxolitinib, which had data from phase 2, phase 3. The phase 3 readout was just out, and we'll be making a major presentation on the oral conferences this summer, so be watch out for it. And then we will hear also some from about the regulatory, about all of these molecules moving in the space. So I would definitely stay open and watch out on these ones, and I'm so happy to get your questions.
That was wonderful and so incredibly informative and very exciting to see all the incredible work that's being done. Go ahead and switch my screen here, but now is our time for questions. So if anyone has questions, feel free to drop those into the chat. There are some in here, so I'm just going to go ahead and start us off. First up is, do you have to be quote unquote high risk patient or have failed treatment to participate in the CALR clinical trials? I think this is specifically asking about Inca. Um, is a patient's VAF taken into consideration, and in terms of molecular response, has it been similar between CALR type 1 and type 2 mutations?
Wonderful. Yeah. So currently the studies for carlaticulin are in the second line setting, but Inca could be added in ET patients to hydroxyurea, not controlling the disease. So not necessarily being off and failing, but being on hydroxyurea high risk and not having control platelets still make people eligible. So there, I would check eligibility criteria with the local centers, but for ET, that is possible to be enrolled. But normally that would be failure, correct. And then for the allele burden, yes, the carlaticulin burden is basically molecular response. That's what I've shown you. And it is shown that the type 1 would respond faster and deeper to lower doses of Inca currently. Therefore, the current investigation is going with higher doses across and building up a cohort of type 2 on different dosing to really understand the pathology. The physiological reasons why is kind of been already postulated by a couple of researchers with wonderful data showing that those two types respond differently to binding. So the molecule binds very easily and deeply to type 1, maybe not so completely to type 2. So there we may have to have a dosing changes. But with those adjustments and appropriate schedule, we see responses across type 1, type 2, including carlaticulin burden. Again, hard to get data from phase 1s because it was so broad from 24 milligram to 200, 500 milligrams. But the doses 400 above, which are considered higher dose, work very well for type 1 and type 2 patients as well.
Awesome. Lynn, you have your hand up. Would you like to ask a question? I can unmute you if you would like.
Yes, it's in the chat box as well. Just relative, we got a little bit of a late start here, and just looking to figure out how to jump on board with all you're saying relative to where we're at right now.
Yeah. So a recording will be available. I'll probably send a follow-up email out next Monday or Tuesday. So the full recording will be available, and Dr. Masarova was generous enough to share a PDF copy of the slides as well. So
That's
those will
amazing.
be accompanying the email as well.
Can you see the highlighted question in the from Lynn? My diagnosis is post polycythemia vera myelofibrosis, currently taking a combination of hydroxyurea at 1500 twice daily, 750 twice daily, 1500 total. Feeling great, cared for at Mayo, aiming toward qualification for a marrow transplant. How might I benefit from these findings?
That's a wonderful question. Currently, we do not know, but there has been, of course, it's going to take us a couple years, but there has been already shown the better the disease control prior to the transplant, the better the long-term outcome. So that just says if people are able to engage in a research that has ability to go after the clones, either it's carlaticulin or hopefully emerging JAK inhibitor, they are more selective, and get those decreased, then one can just postulate the stem cell outcome would be better. But even with the current approach, if people enter stem cell transplant in a disease control, the outcome is fantastic. So it's an only cure. So that's why we would use it as a tool to decrease the disease burden and improve the control. But right now, because we don't have a mature data, we would not postpone or prioritize any investigational therapies over curative stem cell transplantations if people are already having a donors and are set up for it. That doesn't tell that maybe in the future we will be able to basically sequence, right? Either get the best control prior to transplant or use it as a prevention of relapse or control of if there is any disease left after transplant to really get the deepest cure possible.
Yeah. Wow.
That's exciting. Thank you.
Of course. And our next question is, I believe, asking about a hemoglobin level. But is there a reason that doctors may wait for a hemoglobin of 10 if an individual feels terrible being at a level of 16 and a half? Just what is the reasoning behind waiting to start treatment?
That's a very valid and fantastic question. And I think there has been always not really clear-cut definition of either the levels or the response levels. And that has been evolving over the years for different diagnoses. Historically, for myelofibrosis, it's been set up at 10. With the novel prognostic scores, it has been more for severity, so 8 or less, or 9 or less for different genders. But there are also data coming from different diseases across, like myelodysplastic syndrome, where 7 is considered to be more beneficial. So that's an arbitrary cutoff, which has been agreed upon and used broadly, but it's evolving. For example, last year, the myelofibrosis group and Mayo Clinic from Teferi has basically reclaimed and recleaned the response criteria for anemia that has become more consistent and mean to basically decrease or improve by certain amount of hemoglobin levels to be called responses, where before in all the clinical trial development, they were all back left and right. So that is the reason why even some therapies that look very promising may not really make it at first run, because the criteria for transfusion initiation and maintenance vary very broadly across places, institution, and country even more, where some people would not even transfuse with hemoglobin of 6.5, but some people would be already scared of 8. The only setup rules are for using anemia stimulating agents when the hemoglobin reaches over 11 or 12, because that has been in studies for different patients connected with adverse outcome in terms of it could lead to more strokes or uncontrolled blood pressures, because it's just robust response. So that's why those were capped at 11 or less. But in terms of 10, 8, 9, 7, those are very broad. So it's always going by patient's tolerance. If people are asymptomatic, then the number itself does not matter. But if people are symptomatic at 10, that is usually something else going on. So there we have to more do investigation about why would somebody who is otherwise relatively healthy be symptomatic at 10? Because people, if it doesn't happen quickly, usually tend to do pretty well with 9-ish or even 8. But if they happen quick or are driven by something else, then even 10 could be an extreme burden.
All right. And then next up, we have, why isn't ropeg interferon first-line treatment for ET, since it modifies disease and doesn't just decrease platelets like current first-line hydroxyurea?
That's a wonderful point. I think that's because there were no data so far to show. There are now coming. And I've mentioned that we don't have the data published, but something called EXCEED ET was a study that was also finished last year and has data available, including frontline patients. And the one that I mentioned, the ROB ET from Europe, also showed patients that were frontline and low risk. So there are more data emerging, and I am pretty positive as that came with in PV, because PV, it has an indication for high risk, regardless of line, and then also low risk PV, because low PV study has been already shown. So in PV, it doesn't have line, it could be any line. For ET, because we have only randomized data and clinical trial for second line yet, but emerging data from frontline and low risk as well would be coming. I do hope and envision that the indication will be expanding and the agent might be actually starting to be used earlier. But currently, there are no data available that would justify possible risk, because every medication has a risk, over nothing. But as you've seen, if you follow the presentation and have time to review the slides, comparing to even anagrelide in second line, and as we've known from comparing to hydroxyurea in frontline in PV, the side effect profile at the end of the day is favorable to interferon. It is just hard drug to manage. It's not one prescription to go, it's hands-on to find the right dose for individual patients. And that could have been the biggest hurdle by now not to be used over Hydrea, because everybody is easier to just prescribe one drug, don't really care about deeper. But I see more data coming that it has a benefit on disease modification, and behind, it'll be more pushed into alternative over the current standards.
Okay. And then next up, this is asking about another clinical trial. This one is being the ATX-011 from Alethio Therapeutics. Just if you have any familiarity with that one.
Yeah, but I haven't seen any data yet, so I don't think there was anything publicly available that I can speak of. Any new clinical trials that would have an more kind of advanced molecule that would have exciting features behind what's available are very encouraging to me. I like that. But currently, there were no data that would be made public that I can speak of this one.
All righty. And then next up, is there currently another alternative being used instead of hydroxyurea for ET, specifically with a CALR-1 mutation?
Well, except for the interference that we discussed about, there is not really much of, unfortunately, alternatives. There's nothing specific for calr-targeted type 1 mutation except for the clinical trial with the antibodies that we discussed. So not really anything specific that would be used for that typical reason. Of course, there are medication like JAK inhibitor off-label that could be used for symptoms, but that's not typically designed or given for type 1s. I think the differences between the types 1 and types 2 and approaches would only come from the targets of calr-targeted, where because we have a drug that goes against the clone. Any drugs by now did not have any discriminatory power to target type 1, type 2, JAK, or even Maple or anything else.
All right. And then next up is, does the INCA study benefits depend on dosage, or is it a combination with another drug?
Yep, wonderful. As I saw, if you recall the slides where I showed the ET responses with the platelets, that I was elaborating about how quickly the platelets drop and stay down, that was on the high dose. So 400 and above has been considered a high dose, and there the responses were faster and deeper, and that basically led to ongoing investigations on the next phase studies of that dose. So the 25 to 400 is considered low dose, would probably not be pursued, only if introduced later for some sort of maintenance or something, if so. So the doses, yes, it does matter. The types, as we discussed, type 1 versus type 2 also already shown a difference, where type 1 would easily respond to lower doses and faster. Type 2 a little bit more stubborn and need higher doses and faster to get better. Combinations, the only data that we have was from the combination add-on to ruxolitinib in myelofibrosis, as I showed you on the far right side of the slides, that there was a study run in Australia that had 22 patients that add INCA to JAK inhibitor, but it's really a little hard to extrapolate data because those are patients not already doing well. So any therapy added to it that improves upon the behavior of the treatment is actually very positive. But whether that would work better than the monotherapy, which were failures, is currently to be determined. I think there is different safety profile because added to something else could worsen some of the side effects of both of the drugs because they could potentiate. But that's something to be investigated. I'm hoping down the road we'll have different combinations that has not been explored yet, even with different compounds. But right now, the dose is to be determined and the higher appears to be more effective.
All right. And I guess more of a general question, but I know a lot of patients, particularly those with myelofibrosis, may be weighing the idea of a stem cell transplant. How do you think that these new therapies that are being developed right now, how do this stem cell transplant play in when we're looking at potentially disease-altering therapies? Like the INCA trial looks incredibly promising. Do you think that there will still be a place for stem cell transplant?
Yeah, that's. I'm hoping we will learn this decade because that's the era of targeted therapy that started. Right now, we don't have any long-term data of any of these novel therapies. We do have no understanding about more complex diseases, right? Some diseases don't only carry the mutation; they have already other mutations that come along and are there. There is inflammation that is already been happening for 20 years. How does that play for disease to be progressing despite us eliminating the targets? That's all open and unknown. So currently, as I said, would be the best way to sequence, to really try the novel molecules to the disease decrease the most. For example, eliminate the calr-targeted completely or even get it into the lowest burden. smaller the spleen, get better the symptoms, get people in the best shape, and secure it with the transplant to get cured. Of course, if somebody is out of age stem cell, we'll have to just stick with the novel therapies. But if somebody's 40 or 50 and has a donor and stem cell lined up, currently we don't have enough evidence to really only rely on the monoclonals or on the novel therapies. I would envision this to be a combinational approach. Hopefully, in years, we'll enter an era where we do 1, 2, 3 different at the same time that would be able to completely cure it, because it's a little bit more complicated disease than just one driver. But right now, the best advice to it would be to get the disease into the best shape, which has been historically connected with the longest and best stem cell outcomes, right? There's the best success story of taking the transplanted cells, the best success story of keeping them there, doing the graft versus leukemia effect, and providing long-term cure and lowest relapse free. So patients don't want to have myelofibrosis after the transplant. That means, why was the transplant done, right? So the lowest chances of that happening are with the lowest burden to go into transplant. So far, we only were able to use JAK inhibitor to achieve control of spleen and symptoms, but the clones remained. We have novel therapies. We may implement an approach where we use the novel therapy for 1 to 2 years to clean everything to the best level we could, and then cure it. Whether we could only rely on those therapies right now, we do not know that yet. And keep in mind, transplant is a one-time procedure. These therapies are you start, you go. There's no stop. We don't know, right? So you will have to commit for life to go every 2 weeks to some center to get some treatments, whereas you have a stem cell and you are hopefully cured. So that is something to go in until we learn how to use it, how to maybe downscale and not use it that often, or we have a therapy that is being given once a year versus once every 2 weeks, then I would trade it for. But intravenous, subcutaneous, forever are feasible for years, but are not feasible for life. So right now we are stuck with, even oral pills are not feasible for life because they have long-term consequences. So if we have a cure, we should use it. If 20 years, 10 years from now, we'll be able that anybody who has achieved certain type of response on the monoclonals, the disease basically went away and never returned, then we will replace it with transplant. And I cannot wait to see that.
Yes. And then this will probably be our last question. But is fatigue addressed by INCA or any other studies?
Yeah, I see you guys all have the same questions and the same problems with the disease that we all love to get better. Fatigue is the biggest no and go, where most of the, even in myelofibrosis, the recent double therapies, the combos, failed on improving fatigue. We could start arguing about, is it a tolerance? Is it the side effects? It is just fatigue having something we called a ceiling effect, where we could only improve it to some degree and then we cannot do anything about it. But it is definitely being approached by the novel therapies where it does tend to improve, but it takes time because we have to always factor in the possible side effects, the burden of coming to center, right? Although INCA is given a very promising and very deep responses, people still have to come every 2 weeks. That brings significant fatigue. If I had to go somewhere every 2 weeks after a year, I'll be completely tired of it. So that is a different story that brings in the burden of it. But we did see improvements even with the INCA, with raw bag interferon. I have showed you on one of the slides at the far right, improved symptoms that the study did have, and the publication also has outlined all of the symptoms that are in the MPN scores. The fatigue is one of them, and that improved in about 60% of patients at the year mark. So there is improvement. It doesn't mean it's complete going away, like people will have zero fatigue, but they notice significantly improvements. The same was noticed in an INCA. The same partly is noticed in some of the novel drugs. Definitely JAK inhibitors do it as well. I think the problem is improvements in fatigue reflection of improvements in the disease burden itself, but definitely it's tied to improvement in quality of life.
And for any patients who are out there who are interested, we are running a survey about MPN patient fatigue that I highly encourage you to check out. I'll include it in our follow-up email as well. Just a very good opportunity to make your voice heard and really show how fatigue is impacting you. This survey was made in collaboration with Dr. John Mascarenas, who is another wonderful MPN specialist. So um, the MPN specialists really do care about this sort of data, and they want to do whatever they can to try to improve quality of life. I'm sure Dr. Masarova will attest to that. So if you're interested, it's a pretty quick survey, and we would love if you would check it out. But we are going to go ahead and start wrapping up here. But thank you, Dr. Masarova. This was incredible. Both the presentation, but also you taking the time to answer so many questions. Always a pleasure working with you, and I hope that we can do it again sometime.
Pleasure is mine. Thank you for the invitation. Thank you, everybody, for the engaging questions. I do hear you and see you. We are working on it. It's a long journey, but I'm quite excited as I frame it for the targeted era in MPNs. Finally, let me put it that way. Finally, something we've seen in other oncology is coming and rolling into MPNs, and I am very excited about it. Give us a little time, but keep your voices up. We need to know what bothers you. We need to know where to focus, and we need to keep improving because we are nowhere close to the finish line.
Absolutely. Well, thank you, Dr. Masarova. And before we hop off, I wanted to let everyone know that HealthTree does actually have a Black health community. So if this is of interest to you, please feel free to reach out to me and I will help get you connected with that group. We understand that every group has different needs and different challenges that they face, and we want to be respectful as we can be. So this Black health group is a wonderful space. Val, who leads it, is such an incredible person. So if that would be of interest to you, please let me know or check it out. If you just look up HealthTree Black Health, you should be able to find it. But it's a fantastic group, and we just want to raise some awareness for it. And we do have a few upcoming events that may be of interest to you. On Monday, April 27th, we have a Gather community event for caregivers. So if you're a caregiver or have a caregiver that you think could really benefit from sharing with other caregivers, this is a space dedicated for them. We know that being a caregiver provides its own set of challenges. So we have a space set aside for caregivers to get together and just help one another out. So it's a wonderful group. That'll be on Monday, the 27th. And then on Tuesday, May 5th, we have You're Not Alone, financial help for clinical trials and mental health care for cancer patients. So really going after that financial aspect. And then on Wednesday, May 6th, we have Your MPN Questions Answered, a live session with Dr. Michael Grunwald. So this will just be an hour of Q&A. There's not a theme to it. It's just come with your questions. We'll probably have the opportunity for you to pre-submit some questions for that as well. But these are a great opportunity to just have your questions answered by an MPN specialist. So would love to see you all there. My name is Dalton again. My email is up on the screen. And thank you all for coming and being part of our community. Truly, we appreciate you all so much. Thank you to Dr. Masarova for sharing your time with us. It's always incredible having the opportunity to work with you. And if anyone ever has any questions for me or just wants to chat, feel free to reach out to me. My email is up on the screen here. It's just dalton@healthtree.org. But I hope you all have a wonderful rest of your day. Going to go ahead and close it out now.